Hello everyone. In this short video, we will trying to understand the concept of sense, anti-sense, coding, non-coding, positive and negative strands. Okay. So suppose that, all these terminology used usually at the time of transcription. Suppose that this portion is melted, this portion is here, suppose that it is melted for the transcription process, and this is the 5' region, this is the 3' region, and suppose that this one is the 3' and this is the 5'. So transcription, suppose that this is a lower DNA strand having the sequences, suppose that here is A, then T, G, then C, C, then G, C, then G, A, then T, T, then A, C, then G, G, then C, and C, then G. So this is the complementary sequences. Okay. So at the time of transcription, when the synthesis of RNA taking place, the melting of DNA occurs and which forms this type of structure. This is known as the transcription bubble. Okay. So this is not the part of our discussion. Suppose that in this is the double-stranded DNA where two strands are present. The upper strand, the direction is 5' to 3', and the lower strand, the direction is 3' to 5' direction. Okay. So this is the, suppose that RNA. Here A means the complementary will be U, T means this is the RNA, A, G means, C means G, and this is the G means C, and C means there is the G. So this sequence will be similar with the sequence of the another strand. Suppose that this RNA strand it synthesized from this lower strand, and this lower strand the direction is 3' to 5'. Okay. So this lower strand it acts as the template. That's why this strand is known as the template strand. This is the template strand or the template DNA here. Okay. So this is the template strand on which the transcription or the synthesis of RNA occurs. And this is the template strand. Now the complementary of the template strand, this is known as the non-template strand. Non-template strand. And this mRNA, this is the RNA or mRNA, this mRNA it undergo the process of translation, or it participate in the process of translation. That's why it having some of the sense codes. That's why this strand is known as the sense strand, having the code or the sequence of the nucleotide having the certain sense. Okay. So this coding or this sense RNA, the sequence of this sense RNA, which is the sequence will be same with the complementary strand of the DNA, or the sequence of this RNA will be same with the sequence of non-template strand. Suppose that this is the RNA or this is the having the sense sequence. That's why it is participated in the translation process. So this is known as the sense RNA, and this sense RNA which is the sequence which is complementary with the template strand, that's why this is known as antisense. This is the antisense. And this sequence which is complementary with the non-template strand, or the sequence of the mRNA or the sequence of the sense RNA similar with the non-template strand except U and T. That's why this is known as the sense strand. This is the sense strand. This sense strand also known as positive strand, and the complementary of the positive strand which is known as the negative strand. So this sense strand also known as coding strand, and complementary of the coding strand means template strand, which is always known as non-coding strand. So this is whole about of the template, non-template, antisense, sense, positive, negative, coding strand and the non-coding strand.